Okay, we're going to continue um, with muscle. And the next parts that we're going to talk about are going to really apply to skeletal muscle and cardiac muscle. It does apply to smooth muscle as well, how a smooth muscle contracts, but there's a slightly different mechanism in how a smooth muscle actually contracts. And, a muscle contraction is when the muscle shortens and pulls on either end of its attachment points. In skeletal muscle, you can see right there, that picture in front of you, there's a femur. Doesn't matter that you can identify the bone, that doesn't matter. But you see where the tendon is attached to the bone, the periosteum of that bone, and the epimysium is continuous with the tendon. And of course, the epimysium is attached to the outer part of the whole muscle, the whole thing. So when that muscle contracts, when the fibers contract inside, and we're going to talk about how that happens, when those muscles contract, it shortens, it pulls on either end of the muscle. We call that a muscle contraction. That's sometimes identified by the word action. The action of a muscle means when that muscle contracts, what happens, what moves. And we talk about things that move. And of course, if we pull one bone toward another bone, that's called an action. But we got to get into the inner workings, what's called the molecular biology of how that all works. So you see the muscle coverings in front of you. So now we're going to go on the next slide. We're going to identify some other structures that you have to know about. One thing I want to point out to you. Uh, oops. I want to go back. Yes. So this slide, what I want to show you is here's the whole muscle. That whole thing up there is the whole muscle. Okay. And then we take out one little bit of that. And that little bit is called a fascicle. A fascicle is composed of a whole bunch of muscle fibers all bound together with an endomysium around the whole fascicle. All right, so then we go a little bit smaller and we pull out what we call a muscle fiber. A muscle fiber is this bit down here. Okay, and inside one muscle fiber, just one muscle fiber. And a muscle fiber, you're also going to hear me call it, or you'll see it in the textbook as a muscle cell. This is one cell. I'm going to show you why this is one cell. How can one cell be like that and have all these long structures inside one cell, and that one cell will go up a few centimeters into the fascicle. And you think, wait a minute, one cell? I thought cells were tiny and small. Muscle cells have a weird thing that they do that makes them elongated. And we also call them multinucleated cells. So there's more than one nucleus in each skeletal muscle cell. So this whole thing here is a muscle cell. And if we pull out each thing here, each of these long strands is called a myofibril. Myofibrils are where the action is. This is where the contractions actually occur. So each one of these myofibrils contributes to the contraction of that muscle fiber, and that muscle fiber contributes to the contraction of the fascicle, and the fascicle contributes to the contraction of the whole muscle. Right, so let's go to the next slide. So we have gone microscopic now. Let me back up and show you what we're looking at. That picture in the next slide is one of these things. Right, I'm sorry, actually you're looking at a muscle fiber. You're looking at the whole muscle fiber or muscle cell, as I said before. All right, so now let's go forward. So this is a muscle cell. The whole picture here is one muscle cell. Inside one muscle cell are a whole bunch of these things, myofibrils, right? These things at the end, there's a myofibril right there. This is a myofibril. This is a myofibril. Those are all myofibrils inside one muscle cell. Also inside a muscle cell is a nucleus, but there will be several nuclei because these muscle cells are formed by a bunch of individual muscle cells merging together. Now let me show you how that works. You have a picture in your textbook of exactly how that works. So let me show you that one. Okay. This is a picture. This is right in your textbook. Okay. So you have myoblast. So you're used to this word. This word here, blasts. Blasts means the thing that forms the final cell that you're looking for, right? So myo means muscle. That word, M-Y-O. Anytime you see myo, we're talking about muscle. A myoblast is a cell that will become a muscle cell. But what happens in embryology, this is what happens very, very early in the development of a human in utero. Myoblasts form these precursor muscle cells. Here's a myoblast, here's a myoblast, here's a myoblast. And like cells everywhere, they have one nucleus. That's a nucleus. There's a nucleus. Nucleus there, nucleus there, nucleus there. Great. We have all these individual cells that all have a nucleus. Good for them. But what they do is they do a funny thing. They merge together, right? They do this thing. They fuse to form a skeletal muscle fiber. So these cells fuse together, all of them fuse together, and bunches more down here, bunches more up there. They all fuse together. And of course, when they fuse, the nucleus of that cell, the nucleus of this cell, and the nucleus of that cell all become part of one cell. So here's the nucleus of the first cell, the nucleus of the other cell. And then these cells continue to merge together. And when they're done merging, you get what we call a muscle fiber. So this one long strand, this one long muscle fiber, is composed of a whole bunch of individual cells that merged together and became a muscle fiber with lots of nuclei to represent the number of cells that all merge together. Inside this, so we call this a muscle cell. Now it says here a muscle fiber. Well, it's the same thing. Muscle cell means muscle fiber in this case. So this one long muscle cell has inside it a whole bunch of myofibrils. And it's the myofibril that forms the contractile bits, the contractile element. It's the myofibril that actually does the contracting. And a myofibril is composed of a whole bunch of individual subunits that are put together. We're going to talk about those subunits right now. So now I'm going to go back to the other slideshow that you have. Okay, here it is, right? So this is a muscle cell, one muscle cell with a whole bunch of myofibrils, and it keeps on going down this way and it goes up that way. Very, very long cell with a whole bunch of nuclei, right? Here's a nucleus from one of the myoblasts. Here's another nucleus from another myoblast, and so on in each direction. Okay, so that's why we say a skeletal muscle fiber is the same thing as a muscle cell. They are multinucleated. Now you know why they're multinucleated. They have a sarcolemma. Sarcolemma is a membrane around the whole bunch of myofibrils. So the membrane around one muscle cell is called the sarcolemma. Inside a sarcolemma, as inside any cell membrane, you're going to have cytoplasm, but the cytoplasm of a muscle cell is specifically called sarcoplasm. So that's the fluid stuff inside a muscle cell that is surrounded by a sarcolemma. All right. Many, many, many myofibrils. Each muscle cell will have a different number of myofibrils inside there. Doesn't matter. It could have 5 or 10. It could have 100. Doesn't matter. There's just a bunch of myofibrils inside one muscle cell. So there you go. Myofibrils consist of, they have really two big proteins you have to know about here, and they're called filaments. We say they have thick filaments and thin filaments. The thin filaments, and you'll see that they really do look thick and thin, the thin filaments are composed of a protein called actin. So thin filaments, whenever I say thin filaments, you think actin. Actin filaments, okay? The thick filaments are composed of a protein, another different protein called myosin. Myosin is the thick protein, thick filament. So thin filament is actin, thick filament is myosin. I'm sorry, thin, did I say that? Thin filament. Thin filament is actin, thick filament is myosin. You see it right there. Okay. Now, I told you there are these subunits that are put together that allow this long myofibril to contract. And so you have to imagine there are subunits that butt up to another subunit, and they're all attached together. They're all stuck together. Those subunits are called sarcomeres. I'll define a sarcomere in a minute. But a sarcomere in this picture, a sarcomere is from that wiggly squiggly line there, and I'll give you a name of that in a moment. That wiggly squiggly line to this other wiggly squiggly line here. Right, that your jagged line, or however you want to look at that. From there to there, that is one sarcomere. So there's a sarcomere there. Here's a sarcomere here. There's a sarcomere down there. On this other myofibril, there's a sarcomere this way, sarcomere that way, sarcomere this way. So these sarcomeres are stuck together, one after another. Millions and millions and millions and millions of sarcomeres all stuck end to end in one myofibril. And you're going to see in a moment, that's the business end. This is this is called the functional unit of skeletal and cardiac muscle, is the sarcomere. Sarcomere here, from there to there is one sarcomere. You put them all end to end, a million of them together, and when they contract, each individual sarcomere does its own little contraction. When they contract together, you get a big muscle contraction. All right, there's more stuff in there. There's a stuff called sarcoplasmic reticulum. Sarcoplasmic reticulum is a tube system that carries calcium. It stores calcium, the calcium ion. So calcium ion has a +2 charge. And inside of this stuff called a sarcoplasmic reticulum, and we'll get better pictures of it in a minute. Inside there is calcium. So it's a calcium storehouse, right? Just think this is where a warehouse where calcium is stored and ready and available, okay? In between these sarcoplasmic reticulum areas, in between each one, there is a little kind of a space or a gap, I guess you could say. In here is this thing called a transverse tubule. Transverse tubule, nobody ever calls it that. We end up calling it a T-tubule. T-tubule. They're going to see that this is extremely important in how a muscle contracts and getting a muscle to contract. All right, so again, the anatomy of it, right? There are myofibrils. This whole thing is a myofibril. This is a whole myofibril. Myofibrils are composed of subunits or continuous units of sarcomeres. From that jagged line to that jagged line, that's a sarcomere. Put a million of those together, you get a myofibril. Around each myofibril, it's surrounded by sarcoplasmic reticulum. And remember, that's a calcium storehouse, right? So there's calcium inside there. And then at each interval here, there is a T-tubule. So you have sarcoplasmic reticulum, you have a T-tubule, you have sarcoplasmic reticulum, a T-tubule, sarcoplasmic reticulum, so on and so forth. You get it. Okay. Now there's this thing called a triad. The triad is nothing more than the sarcoplasmic reticulum and a T-tubule and then another sarcoplasmic reticulum. So we say the triad is one sarcoplasmic reticulum here, one sarcoplasmic reticulum there, and a T-tubule in between there. We refer to that as a triad. There's three things. And as it says here, one T-tubule and two sarcoplasmic cisternae. Cisternae just means it's a holding cell. It's a holding place. It's a warehouse. All right, so again, let me orient you again. This is a muscle fiber, one muscle fiber, or as I've said before, one muscle cell. This is one muscle cell, okay? One muscle cell is composed of a whole bunch of myofibrils all jammed together inside the one muscle cell. So here's a myofibril right there. Here's a myofibril right there, myofibril, myofibril. We've taken one myofibril and pulled it out. We pull out one myofibril. All right, so zigzaggy line there, you know that's a sarcomere. That's a sarcomere. Next to that, attached to that sarcomere is the next sarcomere. Here's another sarcomere. And there's another sarcomere, and on it goes. To orient you some more, here's the sarcoplasmic reticulum. Remember, calcium storehouse, calcium inside there. Here is the T-tubule. And then on the other side of that is another sarcoplasmic reticulum. Remember, that's called the triad, right? One sarcoplasmic reticulum here, one here, T-tubule there. There's your triad. This area right there is the triad. All right. Now, years ago, 100 years ago, before we knew this was the way that muscle fibers worked, we used to take samples of muscle fibers and we'd put them under a microscope, right? We would stain them and put them under a microscope slide, and we'd cut them up and we'd look at them. And what you would see is this. You would see areas of light bands, dark bands, light bands, dark bands, light bands, dark bands, and we would look at those things and go, huh, I wonder what that is. I wonder how that works. So at first we just said, well, you know, here's this thing here, and that thing from here, from this line to that line, looks just like this thing in the same way, this line to this line. So the name was given it sarcomere. That's a sarcomere. The line, the squiggly or the hatched line here is called a Z line. Now, as I've said, I'll say this all semester, don't ask me where these names come from. Maybe Z because it looked like a Z. I don't know. But that's what they're called. So this is called a Z line, and this is called a Z line. So the definition of a sarcomere, write this down, remember it, memorize it. The definition of a sarcomere is from one Z line to the next Z line. That is a sarcomere. This is the functional contractile unit of skeletal and cardiac muscle. You should burn that in your brain. This is the functional unit. Right? What functional unit? What does that mean? When this little thing, and these are tiny, they're maybe 20 microns across, when this little thing contracts, that is what we mean when we say muscle contraction. When a muscle contracts, what we're saying is Z line to Z line gets shorter. These two Z lines get closer together. That is a muscle contraction. Muscles contract by one sarcomere contracts, then the next sarcomere contracts. When they contract all together, this whole myofibril will shorten. When it shortens, it pulls on the ends. When it pulls on the ends, you get movement. So there's other bands here, right? So you know the Z line to Z line, here, right? Okay, so there are other places and other things that you need to know. So there's a light band next to a Z line. On the other Z line, there's also a light band next to it. On the other side of that Z line is another light band of the next sarcomere over. When you combine this, the light area on one side of the Z line with the light area on the other side of the Z line, you get what's called the I band. These bands will change size. Some of them will change size when a sarcomere contracts. Some of these will change size. Some of them will not change size. And we're going to see that in a video in a moment. Okay, so here's an I band. An I band crosses the Z line. But from this dark band to the other dark band on the other side here, and that's within one sarcomere, that dark band and that dark band, and then combined with the bit in the middle, if you put those together, we call that an A band. And then in between this dark area and this dark area, this lighter area in between there, that's called the H zone. All right, so here's the things you need to know. I guess one more thing. In the middle of that light area here is what's called the M line. So this thing, M line, that's an M line. All right, so here we are. Here's an actual micrograph of that means somebody's looking at this in a microscope, okay? So you get the same thing. This is a real, actual myofibril that has been sliced apart, put under a microscope, looked at under a certain amount of power. Here is the sarcomere. What does that mean? That means that this is a Z line. This is a Z line. That means from here to here, as you indicate down here, this is the A band, from there to there, A band. Here is the M line right in the middle of this kind of lighter area. The I band is this light area, the light area on that side of a sarcomere, or of the Z line. Here's the Z line, that light area on that side of the Z line, and this light area on that side of the Z line. This is the I band. So I band, A band, I band, A band, M line, and then in between this dark area and that dark area is this little lighter area in there called the H zone. So this is what all these things do, okay? We have yet to get into thick and thin filaments. So remember the thick and thin that we talked about before. What these areas of the sarcomere correspond to. So now we're going to break it down. So 100 years ago, this is all we knew, right? We knew this picture. We looked at that picture and we said, wow, I wonder what's going on in there. Now we know this is what's going on in there. This is a sarcomere. Now let's look at the sarcomere. Here's a Z line. Here's a Z line. In there are what we call thick and thin filaments. The thin filaments are attached to the Z lines. Remember back a couple slides ago, the thin filaments are formed of actin. The protein is called actin. So that is actin, this long skinny protein there. That's actin. And then there's a little gap. Then there's actin over here. And this is actin. And this is also actin attached to the Z line. Okay, so those are thin filaments, actin. In between the actin, there's these big fatter things. These are called thick filaments. Thick filaments are made of myosin. That's the protein. The name of the protein is called myosin. Okay, so in the middle, this M line, that's where the myosin is attached in the middle. So you've got a myosin molecule coming out that way, a myosin molecule coming off this way. They're attached at the center to each other, called an M line. Over here, you have the Z line, and the Z line has actin coming off one direction, and then you have another actin coming off the other direction, right? These are thin filaments. So a thin filament there, thin filament here. They are connected at the Z line. All right, now we know how these proteins are put together. This is a sarcomere from Z line to Z line. Now we got to figure out how these things work. Okay, so this is it. So now we're zooming way in. Right, this picture, what you're looking at right now, is this. You're looking at this little tiny area right there. And what we've done is magnified it. I mean, I'm going around it with the arrow, round, around, 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 around. If you magnify that, we're going to zoom in on that area. Now that's what we did. Zoom in. Boom. This is what we have. Again, to orient you, this is the thick filament. This whole thing, all the way along. Thick filament. Made of the protein myosin. So as it says, thick filament, that whole thing, all of these myosin proteins all put together into one big fat thing is called a thick filament. Okay, now we have these other things. These are the thin filaments. Thin filaments are made of actin, but there's a couple other molecules you also need to know about, and we'll get to those. So that's a thin filament, thin filament, thin, thin. Remember, there's that space in between. Right, so I'm going to go back. Here's the space in between. Here's the space in between. Here's the space in between. So now we know this dark area is where actin and myosin are overlapping each other. And this is the overlapping of actin and myosin right there. This area in the middle, what's called the H zone. The H zone is where there's only myosin, only the molecule, the protein myosin. That means only thick filaments are found here, only. And then on the other side of this dark band over here, this lighter segment in there, that is only actin. So I like to say this is naked myosin in there, in the H zone. And over here in the I band is only naked actin. And where actin and myosin are overlapping each other is right here in that darker zone. So that's the darker area there. We don't give that a special individual name. And then here's where actin and myosin are overlapping in the graphic. And then we go forward and we say, here they are. Actin and myosin overlapping each other here, but not overlapping here. Not yet. Give it a chance. It's going to happen. So thick filaments are composed of individual strands of myosin molecules, all linked together into this unusual format. Myosin has a particular property to it that is called a head group. Right? The myosin head sticks out from the thick filament. So it's like little spikes. They stick out. Extremely important. This is very, very, very important part of this. Myosin heads stick out away from the thick filament. What they're trying to do is they're trying to grab onto an actin molecule. They're really trying to grab onto it. However, actin and myosin really, I don't know how many reallys I can say here, really want to get together. They really are attracted to each other. They really want to stick together. They really want to meet, but they're being stopped from doing that. They're being stopped by these other two proteins that you have to know about. The other two proteins, so you already know about actin, the protein, and myosin, the protein. Great. Two other proteins you have to know about. The other proteins are called troponin. This one, troponin. And the other name is tropomyosin. Tropomyosin. These are all part of the thin filament. So the thin filament is composed of three proteins. Thin filaments have actin in them, troponin in them, and tropomyosin. Okay? These two last proteins, troponin and tropomyosin, stop myosin from interacting with actin. They're blocking that interaction. They're keeping it from happening. But there's a way that we can make it happen. So this is the definition of a contraction of a muscle fiber, right? So you need molecules to interact. You need proteins to interact. Remember, actin and myosin want to interact, but they're being stopped from interacting by troponin and tropomyosin. When they do interact, when the myosin is able to grab on to the actin that it wants and desires so much, once it grabs onto the actin, it will do an interesting thing. That head group will then grab the actin and pull on it. That has a name. We'll get to the name of what that is. But you can think of that is how a muscle contracts. That's contraction. Myosin grabs an actin and pulls on it. And of course, that pulls on the whole thin filament, and the whole thin filament gets yanked toward the other thin filament, and they get yanked together, and the sarcomere starts to pull together. So we're going to talk about how that works. Right? So it results from the movement within the myofibril, inside the myofibril. The myosin head group grabs the actin, pulls on it. Actin and myosin filaments slide past one another, shortening the sarcomeres. That's the kicker here, shortening the sarcomeres. The muscle fiber shortens and pulls on the attachment points. That's how it works. So if you get 1 million, 10 million, 100 million, I don't know how many sarcomeres you need to contract all together to pull on the ends of the muscles to make those ends pull closer together, that is a muscle contraction. That's what it is by definition. That has a name, that whole process. That process is called the sliding filament model. So now, if you could see me, which you can't, but if you could see me, you would see me interlacing my fingers, right? I take the hand, my left hand, my right hand, and I interlace my fingers together. So if you have them interlaced just a tiny little bit, they can hang on to each other. If you interlace them more, if you shove your fingers together further and further and further, they're acting just like a sarcomere. The sarcomere is the actin and myosin, the thin and thick filament in the sarcomere is pulling each end closer and closer and closer together. So just like your fingers are getting closer together, That's what a sarcomere does. And you can see that here. That's actually displayed right there. This is a Z line. There's a Z line. Okay? When this myosin, that myosin right there, I can zoom in. Right? When that myosin head group right there grabs onto the actin, you know it's being stopped from doing that normally, but there are circumstances where it can happen. Right? So that myosin grabs onto the actin, grabs onto it, and pulls it. See the arrow? Pulls it. What does that do? That pulls this Z line that way. Of course, the same thing's happening over here. This Z line gets pulled that way. So they get pulled together, and here you are, right? They're getting pulled. That is a muscle contraction. That's what it's called, okay? So I have to figure out how to get rid of the, oh, there we go, get rid of the zoom. What does that do? Here is the actual act of muscle contraction, sliding filaments happening at the molecular level when you look in a microscope, right? Here's a Z line, C line. These are actin. This is actin. That's actin. This is actin and myosin overlapping each other. When the actin and myosin pull on each other, this Z line and that Z line get closer together, right? See, it's gone in a little bit here. It really hasn't changed much over there. But now down here, it's really contracted, right? This Z line and this Z line are very close together. That sarcomere is a contracted sarcomere. And this one is more of a relaxed sarcomere. That's muscle contraction. That's what it is. That's how it works. So a couple of points over here, right? When sarcomeres shorten, thick and thin filaments slide past each other, slide past one another. That's called sliding filaments. We say the thick filament and the thin filament slide past each other. That's called sliding filament model. So H zones and I bands narrow. What the heck does that mean? Here's the H zone here, from here to here. When you get down here to the contracted sarcomere, there's almost no H zone. It almost disappears completely. All right, so H zone gone. I band. The I band starts here, and remember the other I band goes over to there. They don't show you the next sarcomere over. But you can see, right, here's the I band, I band, I band. It gets less and less and less, and there's no I band here at all in this contracted sarcomere, the bottom one. So the I bands, H zones and I bands narrow. In fact, almost to the point where they disappear completely in a contracted filament, a contracted sarcomere. Z lines move closer together, of course, right? The ends of the sarcomere pull closer together. That's what a muscle contraction is. The thick and thin filaments do not change length. So the length of these things is not different. It's just the overlapping quality. How much they overlap each other, that's the thing that changes. They're the same length as they always were. They just overlap more than they used to. All right, so that means that if a sarcomere is Z line and a Z line, one Z line, the other Z line, the sarcomere is getting shorter, right? It pulls those Z lines together. You put 1 million of those end to end. It pulls on all the Z lines together. They all get shorter. Those are attached to ends, right? The ends of those muscles are attached from one tendon on one end of the muscle to the tendon on the other end of the muscle that attaches to the periosteum, which you already know all about. When those pull together, that's a muscle contraction. The way the muscle is attached on either side of a joint will determine what the contraction of that muscle will do. And it will move a joint in a particular direction. And that particular direction is called the action of that muscle. And you remember way back in the bone, when we were talking about bones and joints, there are certain kinds of joints that can move in certain directions and other joints can move in other directions. And the muscles around those joints will move them in those directions. And of course, you can't move a joint in a direction that it can't move, right? So your elbow is a good example of that. Where the humerus and the ulna fit together, it can move in one direction. Your elbow can bend. That's called flexion. Elbow flexion is when you bring your hand closer to your shoulder. And elbow extension is when you go the other direction, when you're bringing your hand away from your shoulder. That's it. That's the only motion you have there. That particular joint, is uniplanar, uniaxial, has one movement available to it. It can flex, it can extend. That's because the muscles, the bicep muscle and the brachialis muscle deep inside the anterior portion of your upper arm contracts. It pulls on the ulna and the radius, and it pulls those bones, your forearm, it pulls those bones toward the humerus. That's called elbow flexion. It can only move that direction because the elbow will only allow that movement to occur. It doesn't cause any other movement. And it's all because these sarcomeres contract one after another, millions and millions and millions of them. All right, so I'm going to move on to the next slide. We're going to talk a little bit more deeply about how the biochemistry, the molecular mechanisms of muscle contraction occur.